Yeah, definitely a lot to cover today. Awesome. Well, yeah, thank you to everyone that's logged on and is joining today. My name is Ryan. I am the instructor for today's lesson. So I guess let me just make sure. Can everyone see the PowerPoint slides that are being shared?
Yes, we can see.
Awesome. Thank you. Let's see. Okay, so I guess I'll introduce myself a little bit. So yeah, my name is Ryan. I am a PhD student at the University of Washington in Seattle. And my research here focuses on engineering bacteria to try and turn them into different bioproduction platforms for basically converting waste products like carbon dioxide or plastic waste or agricultural waste and using that to upcycle into valuable things like biofuels, bioplastics, and other sustainable materials. And so my own research focuses on kind of developing the tools to engineer these things, and then also actually applying these tools to engineer and produce these different molecules. And so that's why today I'm going to be here talking about how we can control what cells make. I guess I just also want to say, if people have any questions at any point, they can feel free to unmute and chat or unmute and ask questions, or they can pop something in the chat and I have a chat window open. I think Kato will also be helping me out to field some questions. So yeah, I'm excited, but I think we can go ahead and get started. Okay, so controlling what cells makes, this happens at a few different levels. And so kind of the outline that I'm going to talk about today is these different levels of engineering. So the first being engineering hosts. So at kind of the cellular level, how do you pick which cell type you want to engineer for whatever you're interested in? And we'll talk a little bit about the strengths and weaknesses of different hosts for synthetic biology. And then I'm going to talk about how to engineer DNA. So how do you express different DNA in a cell? How do you design the type of DNA you want? And we're going to talk a little bit about insulin, which is something that we talked a bit about in week 1, and try and tie that back together. And then I want to talk about engineering metabolism. So you can imagine stringing together different, like engineering components into a larger system where you are engineering the metabolism of a cell. And I'm going to talk about that in the context of artemisinin, which is another kind of example that I think Heidi gave in the first week of the class. Okay, so starting from the top here, engineering hosts. Okay, so the choice of your host for synthetic biology, it depends a lot on what kind of application you're interested in. So I kind of just first broke this down into microbes. So we think of microbes like bacteria, yeast, and fungi. And then also larger multicellular organisms. So that would be like plants and animals. And, there's some like pretty obvious differences here in that microbes are typically easier to engineer and grow faster than multicellular organisms. So a lot of synthetic biology work is done at least at an initial step in microbes because you can do a lot of prototyping there. They have a lot smaller genomes, and so they're easier to work with in that sense because there are like fewer interactions between genes to worry about. Versus multicellular organisms are more difficult to engineer. They grow slowly, and there's a limited number of synthetic biology parts. So kind of every genetic engineering effort takes longer in them. In addition to that, they have larger genomes. They have several chromosomes. They have different organs and cellular organization. So there's a lot of challenges there, and it requires more specialized expertise to work in. So you can imagine engineering in multicellular organisms would maybe be relevant for people that are interested in engineering different crops for drought resistance, or maybe engineering cancer therapeutics in living cells or disease therapeutics, versus engineering in microbes. Maybe you'd be interested in if you're trying to produce medicines, produce fuels or materials, and you want to make them in a sustainable and cheap way. So getting down a little bit more specific into eukaryotes versus prokaryotes. So prokaryotes is bacteria mostly, and then eukaryotes is things that have a nucleus. So they have different cellular structures and cellular organizations that kind of changes how you organize and work with the cells. So eukaryotes are bigger genome and have organelles. And so humans are eukaryotes, but also baker's yeast is a eukaryote. And so there's a huge amount of diversity among eukaryotes. Prokaryotes is typically how we imagine bacteria. So this would be like E. coli, but also any of, your kind of standard bacteria that you might interact with. So diving into this eukaryote versus prokaryote thing a little bit deeper, this is similar to what I was just discussing previously. But basically, bacteria or prokaryotes, they grow faster, they have smaller genomes. Typically, they only have one chromosome, and everything in the cell occurs in the cytoplasm. So if I go back a slide here, the cytoplasm is basically this intercellular space. So the only basically like separation in the cell is the membrane, which is the outside of the cell, and the cytoplasm, which is like everything inside. So most things are just happening on the inside of the cell. And oftentimes bacteria are very metabolically diverse. So there are bacteria that can photosynthesize, there are bacteria that can grow in the absence of light, in the absence of oxygen. There are bacteria that can grow on electricity. So bacteria really have a very diverse metabolism. And then there's yeast, which is probably the other most popular organism for synthetic biology with bacteria. They're useful for studying applications that you might eventually be interested in porting into an animal or plant model, but you want to start in a microbe first because they're biologically more similar. And so they might be better able to express complex proteins that maybe would not express well in a bacterial cell. They have a nucleus, they have cellular localization, and they can sexually reproduce. So you can do kind of crossbreeding and evolution of yeast through that sexual reproduction of yeast. Also very metabolically diverse and industrially useful, although they grow a lot slower, and so they have longer engineering cycles. So does anyone have any questions? Either you can unmute or put in the chat about in what situation you'd want to use maybe like a bacteria versus a yeast for engineering. Or could maybe people give examples of like what they would want to use one for the other for? So I guess, one example I think could be if you were interested in making maybe a biofuel, you could do that in bacteria because bacteria can grow really quickly. But maybe if you wanted to make some kind of human protein and express it in a microbe, you'd probably want to do that in yeast because it might have a better time expressing that protein. But if there's no questions here, then I can move on to the next section here, which would be engineering DNA. I'm going to talk about this in the context of insulin, which we talked about in week 1 with Heidi. Okay, so we talked a little bit about how insulin is kind of the first success story of synthetic biology, and how biologists took the insulin gene from the human genome and were able to express it in bacteria. And that was able to basically save thousands of lives because now they could recombinantly produce insulin, reduce the price, and that could be used as a treatment for people with diabetes. But I don't know, people might not actually know what it looks like, the workflow to make insulin in a recombinant cell. And so the workflow here is basically from the human cell, you have to extract out this insulin gene or find it from some online database. And from a bacteria, you need a plasmid backbone to put insulin into. Let's see if I can do. Is this doing? Oh yeah, here. Maybe this is better. If people can see the laser pointer. So you need a plasmid, and basically you need to build what's called a recombinant plasmid by putting your gene of interest, which would be the insulin gene, into this plasmid. From there, you'd introduce this plasmid into bacteria, and then the bacteria would then produce the insulin. Yeah, so I see there's a question, what are the characteristics of the plasmid that make it accept a foreign gene? We'll talk a little bit about that. Basically, you have to design your plasmid so that it is compatible with the bacteria that you want to put it into. So plasmids have a couple of different things called, well, maybe I'll talk about them in a few slides. Basically, there are two ways that you can make sure that your plasmid will replicate in your organism. And you also want to optimize your gene so that it will express well in your cell. So I'm going to start here by talking about how can we get the sequence for the insulin gene? How do you know what sequence you actually use for whatever your gene of interest is? So I want to come back and revisit the central dogma, which I think we talked about last week with Stephen. But basically, at every level of the central dogma, there's a way that we as synthetic biologists can optimize what's happening in our design. So replication, DNA replicates in the cell, and we can control how this replication happens by controlling what's called the copy number of our plasmid. So the copy number literally means how many copies of the plasmid exist in the cell at a given time. So a high copy number plasmid would mean that there are many copies of the plasmid DNA in the cell. And so it would mean that the genes in that plasmid are going to be highly expressed because there's so many copies, versus a low copy number plasmid would have few copies. And then there's transcription. So transcription is the process from which DNA is turned into RNA. And we can optimize this through our promoter and terminator strengths, which I'll talk about in a slide. And then there's translation, which is how RNA, messenger RNA, is turned into a different protein or peptide sequence. And you can optimize this through ribosome strength or codon optimization based on tRNA availability. Okay. So when you need to find a sequence for a gene, there's some really useful resources online that can help as a starting point. So one would be the National Library of Medicine through the NIH. That's available at this link. And this contains DNA protein sequence information for millions of organisms. And so if you're interested in finding a gene that does some function or makes some protein, this is a really great place for you to find it online. And all of the information is available free. And then another great resource would be Uniprot, which has protein sequence information and also functional information about these proteins and structural information. So it can tell you where in the cell it's expressed. It can tell you what, like if it's an enzyme, what the product of the enzyme is. It can tell you stability things. And so it can do a lot of the information for kind of characterizing a protein for you without you having to do it. Yeah, there's a great question in the chat here. Is E. coli the only bacteria we can use to commercially produce insulin? No. We can also use yeast. And so people have used bacteria or E. coli or Saccharomyces cerevisiae, which is baker's yeast. And they both work pretty well. The Saccharomyces, I think, is able to express the insulin gene closer to the human context. So it requires less effort after the fact to put the two insulin chains together, but it makes less protein overall and it grows slower. Versus E. coli, there's a little bit of modification that needs to be done, but it can make a lot more of the protein. But there are also other organisms that you could use. So a popular organism for industrial biotechnology is one that's called Bacillus subtilis. And I'll put that name in the chat. And people like B. subtilis because it's really good at taking proteins that are in the cell and secreting them to the outside of the cell. And so if you are trying to commercially produce a protein, having a cell that is really good at kicking that protein out of the cell is useful. So that's one example of another organism that would be of interest there. So from which type of human cell is the insulin gene extracted? You don't actually even need to extract it from a human cell. You can, the DNA is in every cell. It's just maybe not expressed in every cell, but you can also just get the gene from one of these databases and maybe order it to be synthesized. And so that's like, you know, you don't actually need the organism that you are starting with to get the gene from. I don't know if that answer made sense, but you can basically find this sequence information online. Did people know these resources before, or did people know any other resources? Maybe if people could just like do a reaction if they'd heard of either of these things before, or put in the chat if they know another place where you can find sequence information. Yeah, SwissPro is another great option for protein sequences. And I think what's great about these resources is that all of the information that they have on the sequences is available, you know, free information. You would still need a way to synthesize the gene. So you can either have a vendor synthesize it for you, or you can do PCR to synthesize it from some host DNA that you have. But this will get you a lot of the work done. Okay, so here's an example of what a UniProt page looks like. And this is for the human insulin gene. So the kinds of information that this has is things like the protein name, the gene name, and the organism it comes from. So this is the human gene. It'll tell you the amino acid length, so the size of the protein, and it tells you how well annotated it is, which means like how much do we know about this protein? How well studied is it? So this summary information exists at the top of the page. And then at the bottom of the page on UniProt, they have this sequence data. And so this is the actual amino acid or protein sequence for the insulin gene. And you can see this was last updated in 1986. So that was like around the time where they first did that recombinant expression of the insulin gene. Yeah, so there's a question in the chat: how do we detect the gene from human, which codes for the insulin synthesis? So historically, I think that they probably did this by studying kind of like genome level things in humans and removing different proteins and seeing like what the effects were. But now we've known for, close to 40 years what this is. And then there's a question, do we get the plasmid from any bacteria? Usually you would start from some plasmid kind of as a template that you can get. It doesn't necessarily need to come from any bacteria because the DNA is, you can isolate the DNA separate from the host that you are extracting it from. And so there's different plasmid backbones or different plasmid types, but it does not need to be specific which bacteria it comes from. Okay, so I talked a little bit about codon optimization. Maybe in the chat or someone can unmute. Does anyone remember, I think it was talked about last week, what codon optimization is or why it's important? Well, I see a hand raised. Michael, do you want to answer? Okay, Michael, if you want to weigh in, you can, but oh, can't unmute. Interesting.
Then you can try it again. I think I can unmute.
Okay, well, if you want to answer in the chat, you can. Not sure why you can't unmute. But essentially, different cell types. Okay, Michael's going to answer in the chat. Different cell types have different tRNA availability. So tRNAs are what recognize different mRNA sequences and tell the ribosome what our amino acid to use. And so depending on the tRNA availability, you want to change the codons that you use in the cell depending to basically optimize your expression. So this is a free codon optimization tool that you can use for E. coli, but you can codon optimize for expression in all kinds of different bacteria or in yeast or even in human cells. Yeah, so Michael's answer here is there's codon bias for every cell lineage, and codon optimization enables us to select the best codon for cloning chassis to use. And that's exactly right. Basically, the codons that are available or prevalent in any cell is going to be different. And so this allows us to basically optimize our chances of expressing. And so this here, this sequence is the amino acid sequence. So different amino acids for the insulin gene. And then you can plug it into this web tool and it'll basically optimize it at the DNA level. So that you will have the DNA sequence that gives you the best expression of the gene that you're interested in the organism that you're interested in. And so this here is a codon usage table. And so these are basically relative percentages of codon usage for different amino acids. So if you're looking here, V is valine is the amino acid, and there's different ways that you could make valine, but the percentage from which they're available differs. Versus methionine, this is just 1.0 because there's only one way to do methionine. Okay. So that's how you can get the DNA sequence for insulin gene. but now we still need a way to express it in E. coli. And that means we need a way to, put it in a context that our cell can express it. Okay, so let's see. Sorry. There's obviously a little broken here. So basically, we want to know how we can go from DNA to protein. And so every RNA transcript is expressed from what's called a promoter. So does anyone know what a promoter is? I guess, yeah, if people know what a promoter is, you can put in the chat. I'll be reading Dan's question here. Yeah, so when optimizing the codon usage, do you consider the desired percentage of GC content? Definitely. So different organisms, different cell types have different GC contents in their genome. So I think E. coli is around 50%, but some different bacteria have really low GC contents, like 30%, and some have really high, like 80%. And so depending on what what the genetic background of the cell you're using is, that would change the GC content of your codon optimized construct. Okay. So I don't see anything in the chat yet about what a promoter is, but promoter is basically how the RNA polymerase knows to sit down on a DNA transcript and start turning DNA into RNA. Oh yeah, so it looks like Dito has a great answer here. Upstream region of a gene whereby the DNA polymerase binds so as to start the transcription. Yeah, the only caveat here is I would also say it's the RNA polymerase. So this is where we're actually reading DNA but making RNA out of it. And so this promoter affects the amount of RNA that's transcribed from a DNA sequence. And then at the end, we have a terminator, and basically the terminator tells RNA polymerase when to stop making RNA. Okay, so the choice of promoter, no worries. The choice of promoter, affects the amount of expression that you get of your gene. And so depending on the strength that you want, you would tune your promoter choice accordingly. And so there's two types of promoters that people typically use in synthetic biology. One is this idea of a constitutive promoter, which is basically the idea that these promoters are at a fixed strength. Oh yeah, there's another answer here. Promoter is a region that contains a specific sequence whereby the RNA polymerase binds in order to initiate transcription. Yeah, that's a great answer. So basically, if you want to be able to control the amount of a gene that you're making in a cell, you can change the promoter strength accordingly. So this here is basically a characterization of a bunch of different promoters of different relative strengths. And you can see there's one here that is the strongest, and you can kind of get expression at every level along the way. So can anyone think of an example in which maybe you wouldn't want to express something as strongly as possible for a gene? While people are thinking about that, I'll talk about this idea of an inducible promoter. So while constitutive promoter is always on at kind of a constant level in the cell, inducible promoters are promoters that you can turn on or off in response to different inputs. And so oftentimes there are certain molecules that will either activate or de-repress a promoter and allow you to go from something that is like very off and then turn it very on to have different levels of transcription there. Are there any questions about constitutive or inducible promoters? Okay. I'll move on here. Okay. So once you have a promoter and a terminator. Oh, yeah, great. Okay, so Dan answered this question to avoid metabolic burden. And that is in response to why you would maybe want to use a weaker promoter or even an inducible promoter. Yeah, there's this idea of metabolic burden, which is basically the cell can only do so many things at a given time. There's a limited amount of ribosomes or RNA polymerases in the cell. There's a limited amount of energy that a cell has. And so if you want to not burden the cell, then you can use a weaker promoter. Also, yeah, reducing a toxic byproduct. Sometimes we try and make cells produce something that is toxic or otherwise harmful to the cells. And if you have them make a little bit less, then you can actually maybe get more out of it by not killing your cells while they're making it. So those are both great answers. Okay, so once you have a promoter and a terminator, you need what's called a ribosome binding site or an RBS. And so basically every mRNA that encodes a protein contains an RBS. And this RBS affects the amount of protein that's translated from an mRNA transcript. This is kind of an important part and across all parts of biology. In eukaryotes, there's also what's called UTRs or untranslated regions at the 5' and the 3' end of an mRNA. And those are also really important in regulating the amount of protein that's made from an mRNA. And then at the end of your mRNA, you have a stop codon that would tell the ribosome to stop making protein. So are there any questions about this idea of controlling translation with the ribosome binding site or with UTRs? Okay, cool. So let's see. Yeah, so we've talked a little bit about how to get the insulin gene DNA sequence, how you can optimize it to express in this recombinant plasmid. But now I think this was a question that came up a lot in different ways. You know, what is this plasmid? Where does this come from? And so plasmids are basically how you introduce DNA into a cell so that it can replicate. Okay, so Michael's asking, could you add both a constitutive and inducible promoter upstream of the RBS? You could potentially. So it kind of depends on what your goal is, you know, why you would want to do that. Usually you want to use an inducible promoter so that you can tightly control when transcription is happening. And if you have a constitutive promoter, you might lose that ability to control it. Although if you are just trying to express a lot, you could have a strong constitutive promoter and then or a weak constitutive promoter and also induce it. And you know, a RNA polymerase can start transcription at anything that it recognizes as a promoter site. So there's not always just one promoter for a gene. Oftentimes there are 2 or 3 different promoters of different strengths in a gene in a natural context. And so you could stack them up if you want to just, you know, increase the amount of RNA polymerases that are being recruited at any time. Okay, so coming back to plasmids. So plasmids are the easiest way to express a gene in a cell is to clone your gene into this plasmid. And so what is a plasmid? It seems like people have some familiarity with this, but plasmids are basically small circular pieces of DNA that replicate independently from the host chromosome or genomic DNA. And so this DNA is not a part of the cell's genome, but is still in the cell and replicating. And so there's some important parts of the plasmid for synthetic biology applications. One is this gene of interest. So this is whatever you're trying to put into the cell. You have your promoter and your gene. And this is really the thing that you are interested in as a synthetic biologist. But then there's a couple of other parts that are important for making sure that your plasmid is taken up into the cell and maintained by the cell as it grows and divides. And so one would be this origin of replication here. And this is what controls the copy number of the plasmid. So how many copies of the DNA are in the cell at any given time. And then the other would be some selection marker. And so this could be antibiotic resistance or something like that. That basically makes sure that the cells that have your plasmid can grow in a condition where other cells cannot. And so if you have a plasmid that gives maybe allows your cells to make some nutrient or resist some antibiotic that other cells that don't have your plasmid can survive. Now you can use that to select for the cells that only have your plasmid, and only those ones will survive. And so that's how in synthetic biology you can control for what you were trying to build. No questions about that? Okay, so I just want to talk quickly about plasmid DNA versus genomic DNA. So as a synthetic biologist, you could also introduce heterologous or foreign DNA through genomic integration instead of a plasmid. And there's, big advantages and disadvantages here. So the major advantage of putting your DNA into the genome is that it is more genetically stable than plasmid DNA. So you don't need a selective pressure to maintain it in the cell anymore because it's now just a part of the cell's genome. And the major drawback here is that it's more labor intensive than plasmid DNA. And I think we will talk more about this in future lessons when people talk about different workflows in synthetic biology. But basically, it takes a lot longer to put DNA into the genome than to just put on a plasmid. Okay, so we talked a lot about how to engineer DNA and in the context of insulin, so how you could put insulin on a plasmid and have it replicate in E. coli. And now I want to talk about engineering metabolism. So how can you go from just expressing one gene to actually having kind of a larger control over the cell? Oh yeah, great question from Tito. How can you assure that the plasmid has incorporated your target gene of interest? So there's a couple of ways that you would traditionally do this. One would be if you have access to DNA sequencing, you can purify your plasmid back out from the cell, or even just send a cell directly to a sequencing service and have them sequence the DNA. And that way you can basically make sure that your gene has made it into the cell and into the plasmid. If you don't have access to sequencing services, the best way to probably do it is through PCR. And so if you are concerned about whether or not your cell actually has the gene that you want, you can basically try and amplify a PCR product that would go through your gene of interest. And based on the size of the PCR product, that'll confirm whether or not the gene is in the plasmid. Are people familiar with PCR at all? Does that workflow make sense? Oh yeah. Yeah, so selective markers is a great way to make sure that your plasmid has made it into your cell. But then there's also like this question of how do you know that the plasmid that is in the cell actually has your gene? And so these are kind of two layers of making sure that your cell has what you want in it. Yeah, great questions, great answers. Thanks everyone for being involved. Okay, So I'll go forward here and talk about engineering metabolism in the context of artemisinin. Okay. So yeah, so we learned basically how to design let's see, there's some questions here. Yeah, so there's a question on, what's the percentage of success? It really depends on the kind of cloning workflow that you're doing. And so I think with pretty standard workflows, there's a high percentage of success. And there's like some common troubleshooting and common failure modes. And I think that ICE is going to talk about that in maybe week 5 when we're talking about what does molecular biology look like or synthetic biology look like in the lab. But there's a lot of kind of tips and tricks you can do along the way to improve your chances of success and also measure your success that we'll talk about. And then Michael's asking about the use of biobricks. So biobricks is basically like a standard synthetic biology part, like promoters or terminators. And then also biobrick has like different plasmid parts. So you can use biobrick compatible components to basically make your cloning easier and more standardized. And that will increase your chance of a success. And so let's see, if we go back here, these promoters that I'm showing here are different biobrick promoters that are all developed by the iGEM community. Meaning like this is kind of community developed promoters that are available publicly. And so if you like Googled any of these numbers and promoter, you'd be able to find the sequence. And so you could know that you're using like a strong or a weak or a medium strength promoter, which would be helpful if you're trying to like standardize your parts. Yeah, good questions. Okay. So we've talked a lot about how to make a single transcriptional unit. So basically going from, making one gene at a time with a single promoter, single RBS, which is really nice if you're interested in making a protein. So insulin is a protein product. But what if you're interested in making a molecule like artemisinin, which is the anti-malarial, another great synthetic biology success story. So molecules are made from different enzymes, and enzymes are a type of protein. And basically these enzymes convert one molecule to a different molecule. And so you can string these together to build a metabolic pathway that can convert, some starting molecule into something that's more valuable or more useful. And this is what we call like metabolic engineering. And so, this is really how we're going from genetic engineering, which is maybe just making 1 or 2 changes at a time, introducing a gene, maybe taking a gene away, to really doing kind of cell level engineering. And so some of the applications here would be different materials like bioplastics, biocement. People have made spider silk outside of a spider cell. Different fuels, so people can make ethanol or other like alcohols or long chain fatty acids that can be used for jet fuel, car fuel, any of these things. Pharmaceuticals, so isoprenoids and terpenes, like artemisinin. These are common drugs that would be found in plants or different organisms. And then food, so you can make different organic acids, proteins, vitamins that are all like important for nutrition. And any of these, are really common targets for metabolic engineering. So this is from this paper back 10 years ago that was making artemisinin in Cerevisiae, so baker's yeast, Saccharomyces. And you can see here that they're starting from acetyl-CoA, which is basically, it's okay if you don't know what this molecule is. It's like a very important molecule in metabolism because it is essential for growth and it feeds into a lot of essential metabolic pathways for keeping the cell replicating healthily. And you can see there's a bunch of different transformations that they're doing here. So all of these green words are different enzymes that they are overexpressing to get all the way to artemisinin. So they're overexpressing 14 different genes to change the metabolic flux. So they're getting more enzyme, more product turnover. And then they're also repressing 2 genes here that would take this intermediate and feed it into a different product that they want to minimize the accumulation of. So essentially, it's like targeted and large scale overexpression and down expression of genes in your pathway or in a competing pathway. And that's like metabolic engineering. And so if you're trying to express a multi-gene metabolic pathway, let's say to turn acetyl-CoA into melavinate, which is one of the precursors for artemisinin, you can do this by expressing a multi-gene metabolic pathway. So from a single transcriptional unit and different RBSs, you can express these 3 different proteins. You could also imagine expressing these 3 different enzymes from 3 different promoters and 3 different terminators. And an advantage to expressing them all from one transcriptional unit would be that there is, basically they are going to be co-expressed. And so you think that they would have the enzymes would be close together, they would have similar concentrations, and there would be maybe like more tight control there. Versus if you express them from different transcriptional units, so different promoters, you might be able to tune the amount of each of them independently, if that's something you're interested in doing. Are there any questions about this? This is kind of a complicated concept. Okay. I'll go on here. Okay. So what are some of the key challenges for building what is like a multi-step, multi-enzyme metabolic pathway? So one is that it's difficult to synthesize these pathways, so to get the DNA sequences and to introduce these sequences into a plasmid. And so maybe this is something that ICE will talk about in a future class, but how can you take large pieces of DNA or separate pieces of DNA and put them into a plasmid? It's also hard to optimize the expression of multiple constructs and identify pathway bottlenecks. And so this can be difficult, but there's computational tools here that can be helpful. And I think this also gets to Dan's question of how do you ensure there's no pathway interference when introducing non-native pathways? I mean, the best way to do it is either just reading the literature and seeing if there are studies on how these pathways interact with each other. So what kind of regulation there is. And that can give you kind of a rational idea of like what interactions might be happening when it's hard to measure. The other ways are a lot more expensive or involved. So you can have computer models of the metabolism, and a lot of these are already published. And so you might be able to download them and use them yourselves. Otherwise, you might need either fluorescent readouts. So you might need a biosensor that is responsive to different intermediates in your pathway. Or you would need like analytical chemistry, which is like really expensive. But that would be like how you would tell what the actual levels of different molecules in your cell is. And that can tell you how things are interacting. But that's like a pretty expensive way. And so I think starting from the easiest way would be look to the literature and see what people have said happens. Then would be use models that have maybe already been published online and try and adapt those for your experiments. And then if there are any available genetic biosensors, that's a great tool. And then kind of as, the most expensive and most involved route would be to do some kind of analytical experiments. And then, the last point here is just that these large pathways can impose metabolic burden and hurt the health of the cell. And so you have to balance making your product of interest with the ability of the cell to grow and replicate. Because at the end of the day, if you're killing your cell, you won't be able to make whatever you're interested in. Okay, so just summarizing here a little bit, what are some of these key methods for genetic engineering of metabolic pathways? So one is genome integration and plasmid transformation, which we talked a little bit about in the engineering DNA section. And this is basically how you can introduce your pathway of interest into the cell. And then the other one would be genomic knockouts. So basically how to remove an existing gene from a host genome. And this can be important. So to Dan's point, if there's pathway interference, if you have an idea of which genes are maybe competing or interfering with your pathway, you can basically delete genes from the genome. And that's a really great way to make sure that the efficiency through your pathway is optimal. And then another way to do this would be through gene repression and activation. So if you don't want to delete a gene, but you just want to make less of the gene, for example, if a gene is essential for the cell to survive, but is also competing with your pathway, you probably can't delete it because that would kill the cell, but maybe you can just express less of it. And gene activation is maybe, the opposite of this, where if there's a gene where having too much of it would kill the cell, you could maybe just upregulate it a little bit. And so one way that people do this, there's a lot of different ways to do it. It's by using CRISPR tools. So there's something called CRISPR interference or CRISPR-I, which will down regulate or repress a gene by physically blocking the RNA polymerase from the promoter. And so you're not deleting the gene, it's still there, but you're going to make less RNA and therefore less protein from it. Are there any questions about this? Okay. So I think that's, oh yeah. So then there's this. So we talked a bit about, how do you go from acetyl-CoA here to melavonate here with these three enzymes. And it's, really kind of intimidating when you're looking at a full metabolic pathway, but there's some, ways that, like, at kind of a base level, this is, pretty straightforward. You're starting from acetyl-CoA here, and you want to go through this pathway to mevalonate. So you want to overexpress or activate genes that are in this pathway, going through these two intermediates. So this is 3 steps, 1, 2, 3, and 2 different intermediates. And then you want to down repress or delete things that would go to these competing pathways. And so these are different methods that you can use to overexpress or activate genes, and different ways that you can downregulate or repress genes. And so this is kind of like what metabolic engineering looks like at that applied level. Questions about this? Okay. So just to summarize everything that we went through today, because I think we went through a lot, the choice of host organism depends on your application. So whatever you're interested in doing, and each host has different tools and challenges that are associated with it. And so there's, not really one size fits all for this. It's application dependent. Expressing heterologous genes requires optimizing transcription and translation components and a way to deliver the DNA to the cell, either through a plasmid or through an integration. And then metabolic engineering can be used to rewire cellular metabolism to produce valuable chemicals. So this can be foods, fuels, drugs, materials. And then there's a whole lot of tools like knockouts, knock-ins, knockdowns, and overexpression that allow us to tune different levels of control over multiple genes and metabolism. Yeah, okay, so that's the lecture. Oh, okay, great. Dan says, when knocking out a gene, how do you minimize the risk of overlooking other potential functions? Yeah, it's a great question. I think, again, I would say looking to the literature and seeing what's published. I think a great resource is ecopsych.org, I think, which is basically a website that has annotations about like the regulation and pathways that different gene products are regulated in. And so you can make sure that you are not also competing other or like interfering with other pathways when you're targeting a gene through that. Okay. So I think at this point now is when we have kind of like open time for working on homework. Is that right, Kato?
Yeah, like maybe if
I can't
if I
hear you. I don't know if that's just me.
okay, maybe if I told you're willing, like maybe to take them through the questions and then they can answer it and submit.
Can
It's
people hear
okay.
Kato, or is it just me? I can't.
I can hear. Can you hear me, Ryan? Can you hear me,
Must
Ryan?
just be me.
So I think Ryan is checking his headphone right now. I have one announcement to let you know about the homework. I mean, you're going to be working on the homework in a bit, but the previous homework from last week was graded already, and you should see the response in your email. I see a lot of like long answers, but it depends whether like you write it down yourself or like paraphrase it a bit or copy it right away. It's okay to like copy whatever is on the internet, but it's better to understand what it means and see what the actual answer is supposed to be. So when you're working on the next homework, I would encourage to like you don't have to like copy the whole thing, but like try to answer in your own words. That's gonna be a lot better. Yeah. Can you hear me, Ryan?
Yeah, I think like my headphones must have disconnected while I was talking for an hour straight. But I think, yeah, I've got it now.
Perfect.
Okay. Yeah, so I don't know. Should I send out the homework?
Okay, Ryan, it depends really upon you, how you want to handle it, how you want to handle it. Maybe if you want first take through the class in your homework, it's okay. If you don't want to take through them and you just send and then they answer, still it's okay. So the approach is yours.
He told me he was on the bus. So
Yeah.
yeah, no, yeah, he's literally on.
Yeah, I think working on the homework is fine. But maybe like the student can take some time to like paraphrase them by themselves. I think that would be great. I'll take off right now to go to another meeting, but like hopefully if you guys have any question, please feel free to drop it through the Discord channel as well, and we would try to like we can communicate right there. I mean, it could be about a homework or like something else. So see you around.
I can stay on for another 25 minutes and help with homework. Thanks, guys. Yeah, okay. To see your question here, gene expression, is it time bound or every gene expression can occur at any time? It is controlled at different times in the genome natively. So some things express when the cell is growing, some express when the cell is fully grown. Some are responsive to different things. So genes will only express if the cell, if the temperature changes or if the amount of nutrients available change. And so there is a lot of different time control over gene expression. And that's, I think, a good reason to use kind of like BioBrick parts or standard synthetic biology parts that are designed to express at pretty much like all times, so that you don't have to worry about when your gene is expressing, unless if you're interested at expressing at a specific time, you can use these different kinds of time responsive or condition responsive promoters to control things. So for example, people use what's called a stationary phase promoter sometimes, which will basically only express when the cell is at it's fully grown, so stationary growth, no longer growing exponentially. And that can be helpful if you're trying to express something that's going to be really burdensome or toxic to the cell, so that it doesn't happen until the cell is basically done growing. Cool. So yeah, the link to the homework is in the chat here. And I'll stay on for another 20 minutes or so to answer any questions that come on.
Okay, maybe Ryan, also still, this same link of the work will still be sent to the email. So don't worry if at all you miss it, but still we shall share it in the email and you'll still have access to it. Maybe before I give back to Ryan, I just have one announcement to put across. I request you keep emphasizing more students to join in for SynBio4ALL Africa, because we have very many, apart from the teaching project, we have very many projects which we are working on to bring up for you. We are working on a project to collaborate with different stakeholders in synthetic biology to ensure that we come in and promote teams which have different projects in line with synthetic biology. So that project will soon come in, but the people who will basically benefit from it, they are the people whom we are sure they have the knowledge of synthetic biology. So please keep on pushing synthetic biology and joining in the lesson. Back to you, Ryan.
Yeah, I also just want to say, I just wanted me to mention that for anyone that submitted their homework too, that those should have been graded and returned. So if people submitted that before the beginning of class, they should see it already. But if people also are still working on the second homework and have questions, they can also ask questions about it here. I see a question. Why are retroviruses able to do reverse transcription? Yeah, it's an interesting question. This is kind of a part of just evolution for them. So a lot of retroviruses have RNA genomes, and that's like kind of a way that they can basically infect cells without the cells recognizing them as foreign DNA. And then they do reverse transcription. So they go from RNA to DNA. And that's so that they can basically put the viral genome into the genome of the cell that they are trying to infect, without it being basically recognized. So this like reverse transcription is basically like a evolutionary hijacking mechanism. But now it's really interesting because people use reverse transcription as a way to study RNA levels in the cell. And so you can use that to basically take a snapshot of how a cell is responding to different environments or different conditions at a given time by looking at the RNA levels. Okay, let's see.
Okay, hey Ryan.
Yeah.
Someone has raised up the hand. I think you can let him to inquire, then we can also answer these ones in chat.
Okay. Yeah, Walter, do you want to go ahead?
Walter, can you unmute and ask your question, please? Ryan, you can be answering the ones in the chat. Okay, Walter, go ahead.
I was saying, there's that area where I didn't speak very well. You talked about obtaining gene sequence from online database. For example, if I obtain, let me say, insulin gene sequence, I will obtain it as DNA sequence from the database, which is just letters. So how would I synthesize this or transform it into a chemical format that I can start now into the plastic? Now that I have the sequence, just how would I transform it into a chemical, something that I can insert into the plastic? Thank you.
Yeah. So I think if I heard the question right, it's basically how do you go from a DNA sequence on a computer to actually getting the chemically synthesized sequence, which is a really great question. There's a couple different ways to do it. So if you have a source for the DNA, so a place that where the DNA already exists and you want to get it out from, you can do PCR. And so you can basically order short primers from a vendor to basically amplify that gene with compatible ways to put it into your plasma of interest. Or if you have the ability, you can get the gene synthesized entirely and sent to you for you to be able to clone it. The availability of these depends on your location. And so I don't know what the options are like in different countries in Africa, but the best companies to do this would either be Twist Biosciences, which I can send in the chat, or IDT technology, IDT DNA. So those are like two companies that we use. Let's see, maybe also what's another one? Those are companies that we use to synthesize for us. I'm not sure how much they send like to Africa, but I think if you have like a lab that you work in, you could ask them if they will ship to Africa. And they basically allow you to you can just type in a sequence that you want, and then they will synthesize that sequence and send it to you. And so if you don't have access to the DNA to do the PCR from, you can order it from them. So those are three companies that could do that for you.
Does this also work with protein sequences, amino acid sequences, or I have to first back translate into DNA?
Sorry, I'm not sure if I heard the question. Was it like, how do you get the amino acid sequence?
For instance, I was doing some projects designed chimeric protein. That was a construct having different portions of different proteins.
Yeah.
And now I was wondering how to come up with chemical.
I see.
Yeah. So I
Yeah.
don't know whether that can be synthesized as well, or must first be back translated into DNA sequence, then the procedure can be done.
Yeah, I think if you have a chimeric DNA sequence, so like different proteins that you're trying to stitch together, it's probably easier at that point to basically order like the gene to be synthesized through a company, because it'll be hard to get the source DNA for all those different genes together. If you are just trying to figure out like how to put the proteins together, I think UniProt is a useful resource there, because UniProt can tell you about different structural parts and functional parts. So it can tell you, for example, amino acids 1 through 100 are this domain, and amino acids 100 through 200 are this domain. And so if you're trying to stitch together proteins that have different parts of different proteins, like that can be useful to basically look online and see what's available. Oh, it's awesome that the CEO of Twist is coming to talk. That's awesome.
All right, thank you.
Yeah, thank you, Walter. It's a great question. Okay, I can go back to look at some of these questions in the chat. Okay, so during transcription, is it all the bases that are changed to be complementary, or is it only thymine that changes to uracil? Yeah, so that's a good question. So it's both. And so because of how the RNA polymerase reads at the DNA, you are getting the complementary sequence, but the T's are getting changed for U's as well. So let's see. I'll like see if I can quickly share my screen here. Okay, so there's just like, here's an example picture. Basically, if you're reading DNA sequence and like you see this top strand here, and if you imagine this is like A, T, A, T, A, T, the mRNA is going to be written basically like matching, or it's using the bottom strand. And so it's going to have a sequence that matches this top strand. Oh, great. Yeah, you can see this part here. Basically, like the RNA sequence will match the top strand of the DNA, but the T's will be swapped to U's, if that makes sense. And so you are getting like the complementary sequence to the bottom strand or the top template strand, but it'll be the same as the top strand. Then there was a question above that. Can you basically throw more light on codon optimization? Yeah, so basically codon optimization is the idea that there are multiple codons that code for a single amino acid. And so I can go back here. Let's see. To like this. So for example, if you had an amino acid L, so if we're looking, if you're looking at this UUA down to the CUG, there are 6 different codons that code for L. And so you have a lot of different ways that you could write your DNA to make an L or like a leucine. But these numbers here are like basically how like many tRNAs exist for this codon in the cell. And so you can see that there are a lot more L's for CUG than any of these other ones. And so if you were trying to make an L amino acid, you would probably want to use the DNA or the RNA sequence CUG to do that. And so that's kind of the idea of codon optimizing, is making sure that you are like using the best codon for each amino acid. Okay. Let's see. Dan says, when knocking out a gene, how do you minimize the risk of overlooking hypothetical functions? So yeah, the best way to do it, I guess, would be a lot of times if genes have hypothetical functions, there are other genes in the genome that can do the same function. So for example, there are like 4 or 5 genes in E. coli that can convert serine, which is an amino acid, to pyruvate, which is just kind of a building block chemical. And so if you are like interested in knocking out one of those genes, knowing if there are like other genes that do the same function is useful. Also, a lot of genes have what's called promiscuous activity. So enzymes do maybe like one enzyme reaction primarily, but can also do some other ones to smaller amounts. So if you want to avoid like these effects, you can basically like on EcoPsych also, you can go look at different reactions. Let's see if I can find a good example. Okay, so I'll share my screen really quickly here. Okay, so this is EcoPsych, which is this website that has a bunch of information about, this is E. coli specific, but they have databases for other organisms. And this is nice because basically you can look at this specific gene, it'll tell you what the enzyme is, and it tells you what the reaction is. So if I'm interested in like this reaction that goes from serine to pyruvate, I can click on this reaction and they'll tell me all of the genes that have this like hypothetical function in E. coli. And so you can like target one of these genes, or you can target all of these genes, and that'll basically, it'll tell you like which genes have this like suspected function. And that can help you like kind of do your like pathway design. I don't know, does that help like shed some light on how you could do that, Dan? Okay, awesome. Yeah, so I think it's about time that we wrap up. I hope that this was informative for everyone and that people learned something. Yeah, the people should work on the homework, try and get it submitted by next week. Kato, what's the best way if people like have questions for them to ask? Is it just to reach out to the email or?
Okay, the best way is for people to join our Discord channel, because for those ones who have joined the Discord channel, have already really and have already appreciated, because their questions are being responded to at a timely basis. If you don't know how to use Discord, it is very simple. If you have Android, you just go to Play Store, Google Play Store, and download an app called Discord. After downloading that app called Discord, then the link for the Discord is there. When it's on your phone and you tap in that app, it directly takes you to our page and you're connected to the Discord page of SynBio4ALL Africa. If you have an Apple phone, still you can go to Apple Store, you'll get the Discord app there and you put it on your phone. If at all you don't want to get an app, you can use the web browser of Discord and still you'll get us there. You just sign in Discord and get a Discord account, then you can start following us. If in case you get challenges, you can send us an email. I will always be there to come in, and maybe we schedule a meeting at any time, and I help you to join Discord. Thank you so much.
Awesome. Thank you, Kato.
Yeah,
Awesome.
also we have a link. We sent an email. ICE created a link where we have all these links to different slides and to different recordings of our sessions. You can also utilize that link. You can also utilize our YouTube channel if at all you need to repeat the session. We have a YouTube channel. You can utilize it. Thank you so much, Ryan. I think you can conclude.
Awesome. Thank you, everyone.